Hi, Melina. Hi, Carlos. Can both of you hear me?
Yep, I can hear you.
Perfect.
I can hear you too.
Perfect. So we'll wait until 9:10. I appreciate you both coming in on time. Really appreciate it. Um, and perhaps we would also take note of attendance. Um, if you don't, if possible, if you know folks, I think actually people by the time it's 9:10, hopefully people will come in. Um, in the
I told
meantime,
Oh, sorry.
no
I
problem.
told most people that the Zoom is on instead, so
That's great. Really appreciate it, Melina. And I really hope if like anytime if you guys have questions, I know it's easier I guess online, but I know they'll make things a bit more challenging for the next few weeks. But it's okay. Yeah, I'm more than happy to help you guys. And it's really something that I find important that I don't want to just leave you guys off to the unknown or like not being well prepped for post reading week material. So hopefully it'll be okay. And you guys will get an idea and a recharge or reset before reading week comes in.
thank you, Mohammed. Really appreciate it.
No problem. No problem, Anita. Welcome, Jacob, Ravati, Sahar. Thanks for coming in. Let me know if at any time you can't hear me. If you also can't speak via mic, it's perfect to also text in the chat like Sahar. I want to ask, how was your term test or midterm yesterday? I know it's a bit tense, but let me know your thoughts. Cheers is here. I also sent a reminder, but you can let others in the team know that we're having our alternative or like our supplementary um, practical today. Perfect. Alta, cheers is here. Cheers. Uh, Great to see you. Okay. Okay. That's good to know. I'm happy that you guys are finding it. No, I've heard from the two of you, so that's a quarter of a population here for now. If there's other thoughts, feel free to also let me know. Could DM, and I don't worry, like I don't mind like the thoughts as well. There were some concerns that were brought up, perhaps of like some specific questions, but overall, I think if you guys were able to understand the material based on what was provided on the slides, and a bit of what Dr. Brunt mentioned, like in essentially the lecture notes, then hopefully you should be good. And perhaps, as often I've been suggested, start thinking about. I know it's very tense, but often when you're right after the peak, you're usually going downhill. So keep in mind that essentially during this period of time, you're eventually going to go back up towards studying once again for the exam. So bear in mind that if you could start pacing yourself, not go down, but rather continue to stay steadfast, moving upwards and understanding the material, and like keeping up with lecture content and the lab material as well, then you should hopefully do well. Um, Okay, we have 10 folks here. Okay, let's continue let's move forward. I'll just wait one more minute. Maybe if you can, just send to your peers, maybe on WhatsApp if you have their contacts. Um, uh, yeah, I'm just thinking, apart from that, let's see. Is anyone at school? Just curious. Today you don't need your lab coat, or at least presently, for the time being, but oh, okay. Not yet. Hope I'll make it easy for you guys. It's really snowy, really snowy. Now I'm actually close by to UTSC, or at least like even though I'm like maybe 15 minutes apart, by drive or like I guess 40 minutes, 45 minutes walking, it's still quite like snowy. It's quite thick, but nonetheless, I'll try to connect some things actually with a bit of the practical today, so you may be surprised. Let's go straight ahead though. Perfect. We have 12 folks, including myself here. Vaisnavi, Alta joined, Fatima joined. Yes, I know some of you that just recently joined know folks as well that are in the lab, probably in your groups. So just let them know once again to attend because I don't want them missing out. And this is actually one of the main concerns I had because of my experience of taking this course, also for another practical this term, but also from last year. And this material is very of week 5. It's dense, but it's also purposely useful for you completing the rest of your term or the rest of the practical for all of us because indeed you have the FLR from the plaque assay, you have the conjugation assignment, the generation time assignment, all of which are connected with this week. So that's why I purposely wanted to have, thanks a lot, Alta. That's exactly why I wanted to make sure you guys are kept in the loop and not, not just limited, but like essentially hindered or jeopardized in your success. So let's go straight ahead. If there's any questions, let me know. I'll be sharing the screen. So you may have to either raise your hand or text in the chat because sadly, it'd be a bit difficult to see you guys over the screen or to see some of your responses. And I'm also recording this the meeting essentially. I posted online if to also serve benefits for us later on. Probably noticing something different on Zoom. I'm trying to make the best use of let's just say Zoom operations. But here you can also probably see the screen. Can I just get some thumbs up if that's the case? Okay, okay. I got two. That's good. By the way, kind request everyone. I know you may not be showing your faces, but please make sure that you're paying attention. Very, very important. And yes, welcome, Chana. Um, anyone else? Okay. Yes. So let's move straight ahead now. And once again, kind reminder, let your folks, your peers know so that way they're also in the loop and not worried and anxious with what they're going to do next week or 2 weeks from now. Because essentially for Thursday practicals, there's an altered let's say, schedule for your exercises, and I don't want you guys missing out or being stressed with time for your FLR following reading week. And I actually want to prepare you guys now or as soon as possible. Before doing so, let's go straight to the exercise and actually practical week 5 material. So I have to make some changes here actually. Okay, so determining GT of E. coli. Notice how actually, by the way, there are specific terms, specific details that are mentioned, like for the organisms, the full name is referenced first, thereafter abbreviations are used, and generation time. Like you're specifying specific details, just like how they're shown here, but even more in your formal lab report. So just bear that in mind with the keywords that you need to include moving forward actually for yeah, just keep that in mind to make some changes. So sorry, some edits here. But essentially, I need also a timer. I just realized. Okay, I'll try to go. And if anyone notices timing going a bit off, just let me know. But briefly, generation time of E. coli is what the first exercise for today's practical focuses on. As well, over reading week, I just want to let you know if you can show me your flowcharts, just send them to me. And like your post lab, pre lab for this week. That will make it easier so that way we don't have to waste too much time next week, especially when we have limited time, or sorry, 2 weeks from now, to look at the material pre-lab. But essentially, generation time is the concept or the principle of being able to analyze, or the I guess the doubling the amount of time it takes for a bacterium or an organism to double in number, like in this case shown. Oh, I have to put a laser pointer. You include that, like shown here in log. So the doubling of number or absorbance, which we'll talk about soon with turbidity. So briefly though, there are four main stages for bacteria or organisms. Generally in bacteria, you have a lag phase, right? So they're getting attenuated, accustomed to their present or their new environment. Sometimes they're placed into, let's say, a new slide or plate. And so they're getting essentially familiar with their environment, and then over time they're maximizing their potential. And this is in logarithmic growth. So bear in mind, it's generally speaking not just linear, but very fast, near exponential. And so they're maximizing the use and consumption of nutrients, metabolites, etc. And so they're growing rapidly from like one colony to multiple, like multiplicity essentially. So they go up and then eventually stagger at a stationary phase, and that's where essentially there's no general growth or no net growth essentially. And then eventually, if you think about the finite amount of size available, let's say for a plate that the colonies are able to grow in, there's limited space, there's numerous colonies, and so there's a limited amount of space, limited amount of nutrients, and in fact, they get depleted even more rapidly. And thus, with that in mind, there's an exponential growth or an increase, generally speaking, of waste products. And so, with that in mind, you essentially are nearly suffocating these bacteria, and thus there's a greater propensity of death, or even just generally speaking, based on this unit here, a decrease in viable cells, those that are alive essentially. Any questions? How's that? Just am I going a bit too fast or no? Feel free to let me know if you find I'm being a bit too fast. Okay. Thanks,
It's
Claudius.
okay.
Okay. Thank you so much. Sorry, I can't really tell who's speaking, who's talking, but I saw Hajra's mention. I'm guessing that was Alta, but forgive me if I'm wrong. But thanks a lot, you guys. Now, really, just to give you an understanding, what would have been done today is essentially you would have E. coli, 5 mils of it, being placed into nutrient broth, liquid broth, like here in these flasks. So you'd have them at 37 degrees. You'd keep them there because essentially the idea is that we're going to measure, and it's going to be mentioned in a few slides, but their absorbance and essentially their viable counts. I think it's better to go there now. Yeah, so as you recall, there's some questions from the slides here, but really I touched upon this one. Perhaps many of them actually I talked about already. In terms of temperature, this is something that we'll even see down the road, but definitely different bacterium and organisms have different different environmental conditions that they, generally speaking, are more optimal in uh, not just reproducing them, but maintaining their metabolism. Um, that being put aside, essentially, there's a bit of calibration required when you're in the lab to actually modify these Spec 20s. So hopefully, I'll be able to show you this, but if not, have a look at this video. Because it is actually very important. And there are instructions shown where the laser is right now in that black like menu option essentially. And so you go through that instruction manual briefly to see, okay, what do I touch? How do I get essentially to absorbance? And the idea is that before you even, record absorbances, which measure the turbidity, i.e. like the density of bacterium in a specific flask or tube, the idea is that we blank our sample first with like essentially liquid broth without any bacterium. So that way it's pretty much you removing and subtracting any background noise that would be present from um, the accumulation of bacterium with the serum or medium that they're present in. Because the serum or medium itself will have an absorbance reading, and so ideally, when you blank, that will essentially kind of like tear in a mass balance to deduct any extra anomalies or any unaccounted and essentially overestimated values. When you start off, and this is something new, but the initial reading should be between 0.03 and 0.05 for absorbance. So that's something to keep in mind. Um, And let's just show once again, actually, I mentioned about logarithmic growth. As you can see, it looks quite exponential. Technically, it's not a, I mean, it does appear to be exponential here. This is called an arithmetic scale, right? Because it's in generally speaking, in uh, the standard numerals, right? Because here, like you can literally have 100, 10 to the power of 2, and then have 200, 300, 400, all in even intervals until 1000. Whereas here in log scale, you're literally going by multiples of 10. So that means essentially equal intervals would go up from like 10 to 100 to 1000. So it's not like essentially you're going up by ones, but here you're going by multiples of 10. So that's something to keep in mind because for not just understanding the material down the road for your lab exam, but more so even just for your assignments, your better understanding of the practical and other future practicals, that you're essentially going to be recording your generation times using semi-log graph paper, which would show you these values like the ones that you see here. Or essentially you would record them using the intervals and increments shown here. And that's what semi-log shows for one axis in log and the other in regular intervals. Um, Okay, let's see. So I'm just gonna skip a few things because there are changes for this term's lab. Specifically, so I mentioned this before, these details, but for part one, something I didn't mention is that your absorbances are 550 nanometers. So if we go up, if you recall, there was that like icon showing the wavelength, also shown here. We actually go back to, so here we actually go to 550 nanometers. So that's something to keep in mind. Um, and then essentially following this, we take 5 mils. So I've placed 5 mils into this and then 5 mils, sorry, 5 mils of your present, like your group's flask into this tube for determining the absorbance reading for your initial count. But then in the microplate reader, that's where these other values come in, including at T0, meaning you're not really doing this manually, which is a unique change. But essentially, that would help condense and reduce the amount of material or condense the material for you guys to explore. Now that's about measuring optical density, turbidity, absorbance readings from the SPEC 20 or absorbance readings using um, yeah, so essentially the absorbance readings. Then the second part, the second option of how you'd be able to record these values involves a viable plate count. And essentially what makes this distinguishable from the previous one is that because turbidity is measured by the cloudiness of a flask, that can also include, debris of the colonies themselves, i.e. they could be dying cells that are present and are actually recorded still. So that would be essentially giving you total cell count, something to note from your lab manual. Whereas for viable plate counting, they only show live cells, or that's what essentially viable refers to. So just bear that in mind moving forward. Any questions? No questions? Okay, perfect. So I'm just going to go straight ahead. Now, the viable plate count method is an indirect method because essentially you're going to wait 24 hours to be able to see these results. In practice, you may want to see these results in real time, maybe in a few hours, which is why SPEC 20 results or microplate readers are more direct. Whereas here for viable plate counting, nonetheless, you still get an accurate read of the true number of colonies. And this is important because you're going to also use these values to determine generation time. And we'll talk about what you need to do. So briefly here, think about last week we talked about serial dilutions, right? So you're going, zero point 0.1 mils at a time from one flask to another. Here, essentially, you'll be, and bear in mind, once again, resuspending well. So that way you appropriately get the right dilution and also the right amount of E. coli because you don't want them to just settle in the bottom of a flask or a tube. So just bear that in mind when you make these dilutions. But essentially, these dilutions will go from 10 to the minus 2 to 10 to the minus 4 and falling zero point 0.1 mil aliquots or inoculations. But bear in mind that here at this stage, you're going to do 1 mil essentially of essentially the 10 to the minus 4 dilution into here with only 9 mils. And you'll see that tube is already pre-made and it looks a bit smaller. And essentially you place it inside there. Um, you can either invert up and down your tubes to resuspend them well, or yeah, pretty much invert them, and then you get that 10 to the minus 5 dilution. And then similarly, you could even go repeat a step. Usually you could do it from 10 to the minus 4, so that way you don't have to, because this is, you're starting with a smaller diluent from the solution here. So you just go repeat the process of zero point 0.1 mils to 10 to the minus 4 to 10 to the minus 6. Now, if well, like considering this in mind, you'd actually thereafter take out zero point 0.1 mils, repeating the same process we did last week for your we didn't do the plaque assays, the plaque assays actually, yes. So you repeat the same process, zero point 0.1 mils, so that would further dilute your factors. Meaning that essentially you would be at 10 to the minus 5, 10 to the minus 6, and 10 to the minus 7. Bear that in mind. That also helps answer those questions that you may see. And that bear in mind, you're also appropriately then labeling your plates, right? With your dilution factor, exercise, group, and the date for all three plates. And they're actually done not only for 10 to the 0 minutes. So essentially, you would have them in the incubator, remember from before, 37 degrees, sorry, at the water bath. Um, I'll just go for it actually. But you would see it essentially you're being mindful of time because you have different incubations that you have to be careful in keeping track of. Because as soon as you know the E. coli that I place into your broth goes in, that's your T naught or T at 0 minutes. So after that, every 30 minutes, you're repeating this process of viable counts and pretty much bearing in mind for with that in mind, really try to be essentially mindful of other components of the practical, which we talked about shortly. But before I move further, uh, the microplate reader, once again, is actually recording these data from the very beginning. And essentially, this column would be of the blank. We have 6 groups. And essentially, you'd be doing 3 reps of 1 mil incubations or 1 mil aliquots into each of these tubes, which can be shown here, or each of these wells. And so each group would go place in 3 by 3, 3 by 3, 3 by 3, or like 3 reps for each group essentially. And I place the blanks accordingly. Once that's after every 15 minutes, it records the data. And essentially, I'll talk about what you need to prepare yourselves for. But ideally, following that, near the end of the practical, I would get those results. And then probably during reading week, we would record the data, or I would present the data to you guys, or the following week. So the week that you come back. Um, any questions? I know 2 more folks just joined or joined recently. Some
So, yeah, I just joined. So what's the plan for the lab we're gonna miss now? So, are the TA is gonna do the lab and send us the data, or are we doing it the week after reading week?
So presently, those are things that, so great question. Maybe I should pause just for a moment for those that maybe, sorry, to clarify this. So because we have week 5, the in-person practical is presently canceled. Essentially, Dr. Brent may modify essentially the exercises that you're tasked to do for the weeks to come. So essentially, it may be a bit more accelerated, which is why, or inshallah, maybe not. It may not be, Allahu a'lam. But I say this because I wanted to prepare you guys, knowing my experience in this practical, that I don't want you guys to be, more tensed out, especially with limited amount of time, limited amount of resources, and the limited and the many exercises. And I don't want you guys being burdened in a way that you can't bear. So essentially, that's why I wanted to have this Zoom meeting, catch you guys up to pace, and hopefully just at least be able to know and familiarize yourself with what's needed, and perhaps reset yourselves, recharge a bit during reading week and afterwards, so that way you don't have to miss out on anything and nor be surprised on what to expect. So, yeah. Answer your question, Carvin?
I think.
Yeah. Okay. If there's more, let me know. But I do want to also finish up the PowerPoint, or I guess actually, if there's any other outstanding questions regarding this, let me know. I'm gonna send them on to the inbox. So hopefully you guys are up to pace with the inbox, right? Like the announcements that I send. So I do plan to send them, but I would not recommend for those that think that okay, like I could just leave or tell my friends they don't have to stay or it's just recorded to just think of it like I would not only the fact that we're all here and that we're able to answer questions with one another, but importantly, you don't procrastinate, right? Because especially now, it's like cut in half of the Zoom meeting from your practical time. So it should be the case that you all pay attention and are able to engage accordingly, and that way you prep up for the 2 weeks to come. But I don't want you to, like delay this in mind, and likewise for um, think the same, or I hope and I expect that you guys take it graciously and proficiently and punctually to bear in mind that we are ahead of ourselves for the future practicals and just even for the deadlines that you have to come. Um, so hopefully that actually answers questions. Sorry. No problem. No problem. And I'm not trying to be tense with you guys. I do want to provide a level of discipline though, because you guys are future leaders. Don't want you guys to slack behind, and you can be really amazing scholars and polymaths. So just bear that in mind. And in terms of direct count though, I'm just gonna move ahead. It takes a bit of time to navigate, but the idea is that similar to like a hemocytometer, you're trying to denote the number of cells that you see in a specific grid. This is called a Petrov-Hausser grid, and so we only have four in our practical. But the idea is that, and don't worry too much, but the idea is that you would use a microscope, determine the cell density based on the number of grids that you have here, and literally count or calculate the OCD, original cell density by the number of cells that you find, right, at 45 minutes and at 90 minutes of your incubation period, and then essentially divide that by the number of squares, multiplied by the dilution factor and the volume that you add in. Now, let's see. You would have added a dye so you would start off with 250 microliters. Actually, I feel like I may be a bit off for time. So actually, I would say simply using the formula here and the results that you know of, or the variables and the values that you know of from the manual. Essentially, this would be zero point 0.5. This would be 500 microliters. And so you would just take these values into account, solve based on the number of cells divided by 16 times zero point 0.5 times 500 microliters. Hopefully, inshallah, you get your values and just remember to convert them back to mils, right? So that's something to keep in mind. You want to have the right ratio because otherwise you may not actually accurately represent the values that you would expect. For direct counting, by the way, it's a direct method. You're seeing generally, it could actually be both dead and alive cells. Live cells are more distinguishable because they look more healthy. They're more depending on, like we know E. coli, for example, are rod shaped, right? So if you see some scrubs or artifacts that are like pin shaped or, think of like the capsule stain or even just like circular dots, you would omit them from your calculation here. Um, Let me just move a bit forward. Watch these videos. Very, very simple actually, and I just talked about the hemocytometer. Any questions, by the way? Anyone? No
I
questions.
have
Okay.
a question.
Yes, okay.
Can you go one slide back? And I tried to count this small red dots. There are 12. It's not 16.
1, 2, 3, 4. No, no. So the are you seeing the squares or the bacteria?
Bacteria.
So for me, great question. So you have 12 bacterium here, yes, but the number of squares is what I'm referring to. So there would be 1, 2, 3, 4. 4 by 4 is
Mhm,
16. So you're essentially counting the number of bacteria within this, within your slide,
mhm,
um, within these 16 boxes. And then that's where you um, essentially include, let's say 12 or the number of colonies or cells that you find, and then you do the
mhm.
calculation accordingly.
Thank you.
No problem. Let me just, I have a feeling I don't want to be delaying you guys. Okay, so imagine we were in groups. This is a bit funny. Because yeah. So essentially, uh, we would go through these details and essentially interpret the data weeks to come for viable counts because they are done overnight. For the microplate reader, they would also come post reading week. So actually, this pretty much ends off most of what I wanted to talk about for just, let me just go out for part one. So you probably saw my announcement. I talked about there being like three different parts. Yeah, part one, which is this, pretty much talking about generation time. Part two is talking about last week's material. We were on time. And then part three is reflective practice and the remaining outstanding questions that you guys may have. So we could save extra questions actually for the end today. But I want to go through what you would have expected from last week and just briefly connect some points. Remember, I talked about serial dilutions. We did it as well last week and connecting that back with material from today. You would have also done your streak plates, right? So you would have seen plates that appear like this as a good example for Micrococcus luteus. So just bear that in mind, that hopefully you'll be able to see it. Like if you have time during incubations or even just it's more of it is a lower priority for because you've already completed this observation, but generally speaking, you would have had a smear here, more distributed colonies with a bit of a smear in your second quadrant, and then in your third one, which essentially would be around this location, because you would have made a T with your second here, and then your third here, you would have generally seen isolated colonies. In their case, they did it more spread out, like in this manner. So that's something that you would have seen essentially individual colonies for your observation and to draw it as figures. Um, Now, I want to go through the plaque assay briefly. I think there are more. Oh, okay. So, with the plaque assay, for example, you would have gotten at least 4 plates. Some of you may have gotten 4 to 5 plates, including your control. So essentially, unlike what you would have expected, and I'm just gonna go to the past because I've teed in the past, and so I just wanted to show you guys a bit of what I've done from the past, so you guys are not like missing out gravely any material modules. Let's go. So as you can see, I'm a very okay, I'm not trying to be boastful, so let's actually continue. But the point is that I really care for your success. I don't like I think of I wanna make sure I do my part. So I wanna make sure that you guys are able to succeed, and I don't want you, to feel lost or confused. So this is an example of what a plaque assay would look like. You notice how like unlike your previous slides is what we have. Let me just show you guys an example. Are you, by the way, able to see the transition? Like you're able to see the screen, right? Everyone?
Yes.
Yes, perfect. So like this is an example of colonies that you would see, right? From your spread plates, for example, street plates. But the opposite is true for your plaque plates, essentially. Reason being is because the bacteriophage is lysing those cells, right, through the lytic cycle. And essentially you would have the opposite, pretty much looking like waffles. But the squares that you see in waffles would essentially be circles that are pretty much holes. So the idea is that you're having many holes indicating your plaques. And those plaques essentially, because you've added them from the mixture that you had with the soft agar into the NYZ plates, importantly that were pre-warmed once again, you want to keep in mind that the number of colonies you see, just like how you've done for different dilutions, serial dilutions like before, the number of colonies that you see essentially uh, with multiplying by the dilution factor or the inverse of the dilution factor will give you the true number of plaque forming units or PFUs. And that's something to keep in mind per mil, right? Because there's importance regarding these units like CFUs, PFUs. For example, let's say you're a practitioner in a hospital or in a clinic in a tropical location. You want to bear in mind that, okay, if a patient, for example, after an autopsy has, or following like following analysis, essentially, you notice that let's say they have 0.05, uh, um, let's say, actually, I want to bring up a better idea. Let's say you actually plated their their serum, for example, like a small aliquot, like zero point 0.1 mils, like what we've done, and essentially place them on a plate, and you found, let's say, 35 colonies in a 10 to the minus 4 plate, right? That's actually a hint, by the way, here because it shows 10 to the minus 3, right? That would have been your first two with lambda, and then times zero point 0.1 because you would have added zero point 0.1 here, which gives you 10 to the minus 4. So you have 35 colonies here. What would be your true density of the bacteriophage, for example, if the person was infected with a bacteriophage? Can you all actually try to calculate that? You have 35 colonies in plate B. How many plaques or PFUs would you expect to see per mil?
You said 32, right?
35.
35.
And I say OCD here because it's very similar. Like, I mean, it is the population density, but I want you guys to answer this question. This can also help prep you guys to understanding how to do this in the future. I'll wait a minute or less. think about what you would do. So like you have 35 colonies, okay? Let's see, I see one from Garvin, okay. Anyone else with ideas? You can also raise your hand if you want to answer. Okay. I have one from Curious. Any other ideas? Or any other responses? If you agree with like the response that you see, give a thumbs up on the chat box as well. But you should be able to know if it is correct. Like I would want you all to actually respond. I'll give 3 more seconds. Okay. 4 zeros. Okay. Yes. So essentially all are alhamdulillah, very good. I think there's one more person that I want to get a response from. If you haven't responded, and by the way, hopefully you're all paying attention, right? In the practical. Still a practical after all, right? So, Basnavi, Samah, Sahar. I know some of you already responded, but I'm just saying their names to remind those that are like maybe temporarily off the screen, that just don't forget to respond. Okay, perfect. So, essentially correct. And actually, it is good to have it in the correct scientific notation. Um, Now, you have it as is. There are different options. For example, how Garvin wrote it down is correct. You can also say 3.5 times 10 to the power of 5 CFUs per mil, or PFUs, because technically these are plaque forming units. So I'm gonna just let you guys know that that's correct. And let's say you had okay. I would want you guys to actually try to practice this out, even in your lab notebook. Just try to come up with other examples, let's say in a range of 30 to 300 colonies. Just choose random numbers, 189, let's say, for plate C, 57 for plate D, 92 for plate E. Just examples for you guys to be able to know and practice. Okay, this is how many PFUs I expect per mil. And the more you practice, the more easier it is for you. Muscle, not just like being able to understand, but hopefully apply. Um, Now that's the plaque assay. Um, let's go to transformation. Transformation, let's see. Just want to read if I have extra things. I wrote something else. I think it should be okay. Okay. Yeah, okay, let's go forward. Bacterial transformation. This is actually very important. In that reason I mentioned this is because you are transforming, and this is a common thing done in the sciences, not just microbiome, but in like medicine, molecular biology, biotechnology, cell biology, etc. The idea is that you're essentially inserting a plasmid or genes of a plasmid into a bacterium to reproduce and to amplify that gene of interest. And so in our case, even though we're doing an entire plasmid, the idea is the same, in that you are using calcium chloride, CaCl2, to permeabilize cell wall. There's also electrophoresis, where an idea is very similar, using a voltage to essentially open and close the pores of cell membrane or cell wall of bacterium, of the plasma membrane. And the idea is that thereafter you insert your plasmid of interest. Sometimes you even have to design them, which is a secondary point, but essentially you have different genes as shown here in this operon, right? For those that remember B12, this is an important point in that you have many genes of interest within your plasmid, and essentially they are arranged to connect with one another, they are arranged to regulate or activate one another at different times, and under different conditions, which is something very important because we have, remember, 4 plates: LB, positive and like with or without a plasmid. Those are two separate plates. One is with arabinose and another is with arabinose plus ampicillin. Sorry, ampicillin, and another is with ampicillin and arabinose. But not aside from that, uh, stepping away from that rather, you essentially place those plasmids in, you stop or you essentially incubate those cells over some time in an incubator, and essentially you get results that would hopefully, as shown in the following slide, following UV, actually fluoresce. And there's actually something to keep in mind with this in regard. Um, Okay, so I want to see this question here. No, okay, so essentially the idea is that you have LB, right? That was your control, and it's in your serum, right, in your medium. Now you have LB arabinose. Arabinose is a metabolite. It is a type of sugar, right, for bacterium that helps them to grow, reproduce, or just even to grow, perform metabolism accordingly through fermentation. But the idea is that you have arabinose. I'm just thinking of another thing. Ampicillin, yes. But I said you have LB plus or minus plasmid. So if you have it with just the plasmid, right, and you like pretty much have this right here, but you don't have any other conditions, and conditions like nutrients that the bacteria needs to survive and grow at least, right? If you don't have the ones that it requires to grow, then like an oxytropic strain, in our case HB101, then you run the risk of essentially these cells not surviving. So what you would expect is that you wouldn't, sorry, let me clarify. In these cells, actually, you would see smears. You'd see like various like thick, large circular smears, similar to this, where there would just be like one big circle, because essentially there are no resistance. The plates themselves don't have antibiotics. So this would be like essentially something you want to take into account for the positive control. You would expect this with the plasmid. However, for the case of your negative control, think about if you would or wouldn't see. Essentially, you would see similar results, or actually, trying to remember. No, you wouldn't. Sorry, the opposite is the case. So essentially, sorry, it's just I had a different setup for today's practical, so a few things just having to rethink about a few things. But essentially, you have the idea that this plasmid, right, has some benefits for the bacterium under certain conditions to grow. In the case of it having like LB slash amp plates, if you have a plate that has ampicillin, the antibiotic, right, of the penicillin family, the idea is that with the plasmid, it should be able to grow. If it doesn't have the plasmid, then it wouldn't be able to grow. So bear that in mind. The colonies that you see in this amp plate essentially are transformed colonies, the ones that are alive, the ones that you or at least see spots and colonies growing in colonial growth, or just growth, essentially dots like these. Those are your transformed colonies, and you can also calculate the number of CFUs. But for transformation efficiency, this is where it's a bit unique in that you actually add in arabinose, shown here for with its promoter. Reason it's important is because as a metabolite, it's actually going to activate this plasmid, in which you're going to have GFP fluoresce or get activated and essentially promoted in gene expression. This is important because you know GFP is going to fluoresce under UV, right, shown in the following slide. And so hopefully you may see a faint greenish color in those colonies, but the idea is that that's what you would expect to see, or that would depict the number of colonies that are actually transformed and have a full plasmid present without any, constraints, generally speaking. Um, gonna just pause here. Let's see if no. So I'm just gonna clarify. Yeah. So essentially, the ones that are transformed with the active plasmid, essentially with arabinose included, um, uh, essentially would show green colonies. In the case of without, like if you have LB plus AMP, those are still colonies. They're still transformed, right? But the thing is that they're not going to fluoresce because the plasmid itself is not activated with arabinose, which is why the fourth plate that you have with arabinose would show a fluorescent green hint or hue. Any questions, by the way, regarding that? Arabinose, yeah. It's actually AR, so you have it shown like this. Just gonna rewrite it for you guys. It is a metabolite, like a sugar, a complex sugar, for like in biochemistry, you may be aware. But essentially, that's your you have four different plates, and pretty much, I hope we've talked about what you need to know about them thus far. No problem. Let's see. Uh, Actually, that's what I wanted to talk about first, or actually second after the plaque assay, but it's okay. Conjugation is something that I think is very important that we sometimes forget to account for, or at least there are different things that you want to keep in mind. Um, for conjugation, you have essentially two different bacteria, one that will essentially be a donor and one will be a recipient. Now, essentially, you have the conjugation pilus that would be present and actually activated to grow in your donor cell, from your donor cell, shown with an F plasmid. It would have to have this plasmid present, or it would have a similar type of plasmid present, and essentially become the donor or be the donor. And essentially, with that in mind, that plasmid itself actually essentially um, delinearizes or decircularizes from being a regular, in this case, double-stranded DNA plasmid or double-stranded plasmid to essentially linearize and enter through that junction, through that pilus, into the recipient. I say this because you want to understand the theory behind going into biology. And so this is important because you have HT99 and J53R. J53R is oxytrophic. And something to bear in mind is that both of them have susceptibility to different types of antibiotics. However, what's unique, and as you would have done these drop spreads, I can even show you actually an example of them from the past. Let's see if I go back. I actually wanted to show it to you for all of the cases. Just give me one moment. So let's go with RF, the ripamycin, rifampicin swab. So you would have made a streak like this. And so you can see J53, J53R. Mind this, but or it could be also a different stream from before, but I think actually it was J53R at the time. And that's essentially showcasing for you that it is essentially able to grow and thus resistant to rifampicin. And then whereas HT99 couldn't grow, which should denote something else, right? So bear that in mind. And then let's compare that now with the chloramphenicol, which we use often in micro and cell biology. So you could see comparatively, HT99 does grow, whereas J53R doesn't. Right? So that's something to keep in mind. But if you have both now, remember let's go back to the slide. Remember, for about 1 to 2 hours, we essentially, I think an hour, 80 minutes, we put it in, essentially in like these two tubes, the first two tubes that we had into this one with a black lid, 0.1 mils of HT99 and 0.9 mils of J53R, two strains of E. coli, into together. They were resuspended, hopefully, and then placed together into a tube with liquid broth, and they were left to incubate, remember, for about 80 minutes at 37 degrees. Now, this was important because this would actually help the bacterium to actually not only grow, but interact and hopefully conjugate with one another. And one would be the donor and one would be the recipient. Think about which one would be the case based on your manual and based on what you uh, understand from the practical and what, or actually what you're about to see soon. So think about that just for a moment. Keep that in mind because after that you pressed or you placed a drop and smear spreaded your bacteria onto the plate. And now let's go back to, sorry. Let's see what you would have seen. SubhanAllah. So you see actually multiple colonies, which you may think it sounds odd, right? Because like, why are you seeing colonies like spread plating? But the idea is the same actually, because these are not like, remember this is conjugation, right? So these are colonies that were conjugated and successfully are now essentially, um, let me just go back. They essentially have both plasmids and or at least both um, antibiotic resistant genes, right? So keep that in mind. That would have meant that one of the bacterium, either HT99, J53R, perhaps both, but I would say one or the, like essentially that colony that you're observing from before would essentially be one that has resistance to both antibiotics, right? So that essentially depicts to you successful conjugation, and then you could calculate the original cell density as well, or at least observe thereby the success of the conjugation experiment. Any questions? Or before I go to questions, um, I want to show you one more page, the control. Because it's good to always have controls, right? Think about why that would be the case. Oh, I have them aligned, so that's okay. This is another example. Um, Actually, this is an idea, question I wanted to bring up to you guys. Think about this for your post lab, and actually, based on what we're talking about today, I want you to think critically about this. So, rapamycin can grow with J53R. J53R E. coli can grow in rapamycin, and HT99 E. coli can grow in chloramphenicol. But here you have a plate that doesn't have, or actually has two, has maybe actually a few. It has a few. 1, 2, 3, 4, 5, 6, 7. Maybe that's eight colonies. But you could see compared to the one before, it has fewer, right? And some of you, like, I want you to think, even hypothetically, like, why is this the case that perhaps you see fewer colonies? Um, Oh, sorry, I missed your question, Alta. Is this a black market? Yes. So students of the past they were counting the number of colonies to indicate essentially success of conjugation. Um, Now the question is, hopefully that answers your question, Alta. Um, the question you may be wondering, okay, why is there this discrepancy or why is there heterogeneity? You know, maybe there's multiple plates have different numbers, right? Think about the things that we talked about. Let's go back to the PowerPoint. There are certain steps in the methods you want to keep in mind. So I talked about, right, the one drop method. That was done successfully, right, for both groups, right, that we saw for the different plates. But now when we look here at the doubly treated antibiotic plate with essentially for the two groups, I want you to think about what could have been done incorrectly or what could have been done better. Perhaps were the starting colonies and their cultures essentially resuspended correctly, right? This is something I tried, like I want you guys to really think about critically, even from your groups of the past, because, you know, if you don't essentially, either in this case of adding in the correct, like correctly resuspending in the right volume into this final tube here, either at that stage or the following stage after 80 minutes of incubation. If you didn't resuspend well, you're only collecting, think of it like molecular biology or biochemistry, you're collecting pretty much a supernatant, right? Meaning that you're not collecting the cells that you actually want to be spreading into this plate in the first place, right? And so after 24 hours, you're going to find a few single colonies present. And that's something that you want to keep in mind. So one is resuspension. Two is the case of the time. We controlled for timing, which should be okay. But the other is even the volumes, right? Because if you made a mistake, if let's say you didn't use the pipette correctly, or you put it in the wrong amount, or there was air bubbles in your tip, right? Or if you, let's say, didn't follow aseptic technique, right? And you or I'm gonna talk about aseptic technique after, but if you didn't put the right volumes, let's say you put 0.01 mils or 0.09 mils of J53R in either case, then you may see very few colonies, right? If you put more, then you should see colonies in if you done the right ratio, right? Or if you did like if you put the wrong amount, then you may expect to see like if you put less of each strain, then you would expect to see fewer colonies accordingly. Think of it like a dilution. The other point is actually of your aseptic technique, and that's something very important. One is that, and actually what I want to talk about more so is the L rod. And you may not notice this, but like when you're doing sterilization, how quickly or how carefully are you in terms of your aseptic technique? Because you have an L rod, you've placed it with, you've flamed it, right? You've also placed like flame and ethanol, obviously not together, right? Flammable, but you essentially followed the sterilization of, or the aseptic technique rather, of flaming your L rod, right? And before this, actually after this, with 95% ethanol, right? And you let it dry, right? Before then, and then following reheating, you wouldn't essentially place your L rod directly into your plate thereafter, right? You would let it cool, right? Otherwise, you would actually kill the bacteria, right? That you grew post incubation. And so that's why you may actually see no bacterial colonies. So I hope I don't I'm honestly not wanting anyone to see no colonies, but those are three different things to keep in mind: the right volume, the suspension, resuspension as well, and essentially aseptic technique. Any questions regarding that? Okay. So, and I want you to think honestly about this. This is something very important, and it would save time for you really to think, contemplate, resonate, or and ameliorate your discussion points for this week's practical for the post lab, or at least for your understanding, right? And I want to say before ending off part two, uh, briefly, actually. Actually, let's go to reflective practice. And alhamdulillah, we're actually good for, I hope, I'll try to be 1 minute quicker. So let's go to, before the reflective practice, general outline for the assignment. This is also for both practicals 2 and 8, because I'm teeing both, alhamdulillah. And I want to let you guys know that essentially, I know this week would make it challenging, but we'll see what we get from Dr. Bunt, inshallah. And I actually have an idea because I have, like, we'll see what, like, essentially, I have results from the past that I could use to essentially show you guys what you would need. The idea is the following, and I'm just showing it as a sample because your groups will be able to include these data more directly. So let me go to these pages that I have. So I'm gonna, like in due time, I wanted to show you guys this in advance so it could help prepare you for the generation time assignment. Remember, you have like, so if we look briefly at these instructions here, I talk about like you're going from 0 to 15, 0 to 2 hours. Every 15 minutes, the microplate reader automatically takes the turbidity readings for the 3 reps that you had, remember, from the 3 wells of each group, plus the blank. And so the example here is that, okay, you have essentially, if we walk through group 1, this is just a sample from the past. Let's say these are your replicate readings, right? And the blank is quite high actually, but hopefully in our case it will be lower because if you recall, it would be 0.03 to 0.05. There's also some things you want to keep in mind, like why could it be the case, not of the sample here, but hopefully of the results you get, or even of the sample, why it may be higher than you expected. Could it be that it was left too long, or actually not left too long, but could it be that, sorry, no, let me clarify, 0.03 to 0.05 for your first count of replicates. So let's do the math. It is likely more, but nonetheless, we'll do just a sample calculation. So if I'm going to calculate the mean of these 3 sets, let's say it's 0. So how I do this is also like, if you think about it, all of them are essentially over 0.25. So you have 11, 0.011, 0.08, that's 0.19, and then 0.04. So pretty much 0.023. Divide that by 3. You're getting like, I don't want to confuse you guys, okay? But the idea is hypothetically, let's say you get 0.254. Actually, I want to make sure I get this right. 0.0, let's say 0.2566, for example, or 67. Now, if that's your mean for these 3 reps, right, and you can use a calculator, you subtract the blank to get the right value. So in our case, it would be 0.0, let's say even, let's just say hypothetically, it's actually probably 0.78. But let's say that was your value. You would repeat the process for all, so all groups would essentially have these values filled in, but you would want to fill in the mean values for these 3 reps across all groups, and then correct them by the blank and putting that value for all groups here. Once you get those values, let's say 0.09, 0.08, 0.03, etc., right? Like ideally within range. But if nonetheless, if it's, you use the values that you have, and then eventually you would essentially get the mean. So now you're getting the mean of all means of the means right here, right? And then you would take that for your time point here at 0 minutes, and then repeat for 15 all the way for the remaining essentially tables to 120 minutes, which is essentially 2 hours. I want to give you guys this intel now because I don't want it to be like, I want you to be prepared for reading week. So that way, if you get the results then, you'd be able to perform this quite early and in advance, and hopefully get marked in advance as well. Now, that's one major component of the lab. Pretty much, actually, once you've done that, remember you're using semi-log paper to essentially, as I mentioned earlier, deduce the generation time. I talked about it there as well, but the idea is that you have semi-log paper, right? Remember the logarithmic growth. So I know you see 1, 2, 3, 4, 5, 6. It goes up like without those numbers, but don't get confused. This is like if let's say this was 1, this would be 1, this would be 10. Think of these like to the power of. So this is like 10 to the power of 1. This is 10 to the power of 2, 10 to the power of 3, 4, 5, 6, etc. Right? So you're going like 10 to 100 to 10 to 100. This is more understandable for viable counts, right? Because you're going to the thousands, 10 to the power of like 3, 4, etc. But the idea is for at least absorbance readings for the microplate reader, you'd be like 0.001, 0. Actually, I would even say start with 0.01, right? And then 0.1, and then it would be 1 pretty much. So just to bear that in mind, These are smaller graphs. Literally, you're going up to like 3 maybe because their absorbance readings are in the minutia. They're in a finite range. Whereas in like the case of viable counts, you saw from like the serial dilutions and the plaque assay, you'll likely see it from your spread plates, or even just for counting from the past number of CFUs, you're going like to 10 to the power of 7, right? Or more even. So you just want to keep in mind that's where these values are more um, applicable essentially, but the idea is the same. And essentially you would take the slope. So there's two options for this. You either take the slope like this, think of it as like y2, like y, if you think of a linear slope, y equals mx plus b, you do a line similar to this and then do the log, because essentially that should look actually something like this. So think about how everything's connecting now. The absorbance readings, the viable counts, you should see something similar, like to a pattern like this, because we're generally speaking in log phase. It may also be just straight, but it may also be at lag and then eventually reach stationary phase. Um, um, Great question from Cherish. I'm just gonna answer it now. Okay, sorry. Forgive me if I'm mispronouncing. But I do want to say to clarify things. Spectrophotometry, you're only taking the initial value. Remember how I said 0.03 or 0.05? That's only for your spec 20. Actually, I'm gonna go back here, not to confuse you guys. So pretty much is just your initial measurement at time 0. Everything else for spec results are done through the microplate reader, and that's going to be generated for you. Like you'll do the analysis following that. So you don't have to worry about recording. I mean, yes, you are doing the recordings, yes, but you're only doing it for spec 20, like this box here, at time 0. So it's very simple. It's literally you just put in one tube with your E. coli, following it being blanked, and then you just record the observation, which is between 0.03 and essentially that range I mentioned earlier. But apart from that, yeah, hopefully that makes sense. This is actually a change from the past. Alhamdulillah, you guys are having it easier because groups, yeah, I don't wanna venture off, but essentially you have four sorry, let me go back. Okay, let me to clarify or to expand. Essentially, you're doing this for both. So let me go back here. Yeah, turbidity results for two temperatures. Our lab has just at 37 degrees. Right? So the other lab that's beside us would have at 30 degrees values, and so they're going to refer their results. Hopefully, something like this, and you would repeat their process for the other time point. So bear in mind, I know it sounds like there's a lot, but I want to prepare you guys, or at least I want to do justice in preparing you earlier and not before it's too late. So the idea is that you do these results across for like calculating the lab average absorbance value for each time point and for each temperature, 37 degrees and at 30 degrees. This could be done like in an hour or less, even maybe 30 minutes. Yeah, like maybe an hour, we can just say for like the tables for both sides, more or less. It's okay even if it's more, but or around an hour or so, likely less. But essentially, there's that component for microplate absorbance readings, and then you have the viable counts, and that's my second one. I don't know if let me go back to the slide here because, oh yeah, I want to talk about how you calculate generation time soon, but I want to make sure slides wise, critical thinking questions. So these are questions you want to also, based on what we're talking about, discuss. Okay, let me just think. Let me see. GT. Okay, for the generation time, so there are 2 points, two methods of determining it. One is from the slope, like I mentioned, you make a line of best fit, and you're going to do that for all of your plots, the two curves that I mentioned here for microplate absorbance for both um, actually, just to clarify it, you're also having like you're plotting those results, right? Remember the lab, like I said, those averages, you're gonna plot them for absorbance here. Absorbance readings OD. Remember the units is OD subscript 550 because the wavelength is at 550. So bear that in mind when you're recording your units for all axes. For the y-axis, it's OD, it's absorbance readings or absorbance values, and you could say mean lab absorbance values, and then brackets OD 550. Well, x-axis is your time, right? So we're going through different times for the different exercises, but nonetheless, you have time in minutes. And for microplate readers, from 0 to 120 with 15-minute increments. So bear that in mind. You do it evenly, and that you generate a line of best fit and that you've labeled that this is your curve with a line of best fit at 37 degrees and another curve with a line of best fit at 30 degrees. You can use the same table, another table, like another sheet, or ideally in the same table, ideally in the same graph or a similar graph. And then you would just label, you could color code if you want, like this is a red one for 30 degrees, this is a blue one at 37 degrees, something like that. But then following the labeling and the plotting of your data, that's where the generation time comes in because from your line of best fits, you could use the slope itself, calculate essentially what the slope is, log y2. So essentially, you're not going to double log because your values. Oh, sorry. Let me step back. Yes, you're logging. Actually, I want to make sure. Uh, let's see. Of your raw absorbance values, you are logging. I was just thinking about it a bit more technically speaking, but you are like essentially doing the log, or essentially you're calculating the slope based on the formula, right? Like y2 minus y1 divided by with the logs, right? In the numerator, log 2, let's say log 2 minus log 1, and then x2 minus x1. And then you essentially, to determine a generation time, you actually do 0.3 divided by the slope. So bear that in mind. You finish with this final point to the solution. The alternative method, that's one main method. The alternative method is essentially seeing where they double, and not double like 2 to 4 because remember these are in logarithms. But let's say you had 0.04, like hypothetically, let's say this was 0.2 in absorbance readings, and this was 0.4 in absorbance readings. So you essentially go from the y2 all the way to intercept that line of best fit, and then go down to the x-axis. And it doesn't show it here, but let's say this is you're going down from the y2 all the way to the x2, and then pretty much let's say this is um, y1 at 0.02 absorbance reading. Then you're going down to the line of best fit and then go down to x1. The difference between x2 and x1 would essentially be regeneration time. So those are two alternative methods. I believe Dr. Brent likes both of them being included, or like they would be ideally used. So use both. Or if we'll see in future discussions. But the idea is that you do that for the microplate absorbance reading, and then very similar for the viable counts. Differences, remember, you're doing the dilution, like you're calculating the dilutions themselves, right? Based on the values that you see. This is the plaque assay, by the way, but the idea is very similar, right? So, like, once again, remember, let's say it's 10 to the minus, let's say you have 70 colonies, 10 to the minus 5, then 70 times 10 to the power of 5, right? You're doing the inverse, like in that in mind. You're like dividing that by the dilution factor. You get 7 times 10 to the power of 6. Remember, I actually went up because I went to 7, not 70. So it'd be 7 times 10 to the power of 6 cells per mil. That's an example. Let's say you go to 1.4 times 10 to the power of 7, because that would be pretty much double. Then the idea is very similar. You would repeat the same process if you have, let's say, let's go back to the same, actually, the same graph. If let's say this was at y2, you go down all the way to x2 from your line of best fit. Repeat pretty much the same process of just plotting with, remember, the mean values. And then hopefully you should be able to um, deduce essentially the generation time. No problem. If you don't mind to share your name, that would be greatly appreciated. Just I'm speaking to the person in the chat. Sorry. But essentially the idea holds very similar. Remember just how I talked about absorbance readings, and for those that are new to joining the group, essentially I'm referring to the formal the generation time assignment, which is going to come up soon. So just preparing you guys well in advance. I talked about also a bit of generation time earlier, as well as the sample results from the past of conjugation, spoken about streak plating, transformation, and the plaque assay for the formal lab report. I didn't say it specifically, but that's actually part of the formal lab report instead of conjugation, or sorry, the plaque assay is important, and then transformation as well is something that will yeah. So, and then I went to essentially going back to the past in week 5 material, talking about preparing you guys for the future in the generation time assignment. But essentially, you're calculating slopes. Let's just see. Saad, okay. Okay, perfect. Okay. Muhammad Saad, Rahman. Perfect. Okay. By the way, please let others know if there's other group members. Once again, I mean, it's advantageous to come as soon as you can, once again, based on the notification. So just bear this in mind, or hopefully keep uh, keep close eye, essentially. Keep pacing yourselves accordingly with the material. But essentially, you do the same process as I mentioned, and details will be discussed later on in due time. But let me just see before I end off. Microplate reader differences the units as well, like the values. You're going like 10 to the minus. Sorry. Let me just go back, or actually forward. The idea is very similar. So if there are questions regarding this, we'll answer them at the end of today. I would say at the end of 10:40. Um, on the same. I'll answer that at the end actually. If you don't mind, maybe to remind me. I realize that we only have 10-ish minutes, so but of course, if there's questions that you guys want to ask, like I don't mind to stay longer, and hopefully you guys don't mind as well. But these are some questions. Let me go back. I need to make sure I didn't miss anything. Okay. I believe that's everything. Yeah, so in total, you have 4 curves. Essentially, the microplate reader at 37 degrees, the absorbance, the lab values, lab total, or sorry, lab mean averaged values corrected right from the blank, OD 550 values for each time point at 37 degrees with the line of best fit, same for 30 degrees, calculate the generation time using this formula, both for, at with 0.3 divided by the slope essentially, or by essentially tracing the doubling of either OD or turbidity, and sorry, either OD or viable counts, and then judge the difference or calculate the difference between X2 from X1. And so that's generation time. Now, in terms of these reflective critical thinking questions, this is what I want you to do during reading week, and individually show this to me as your post lab. You can show this to me as part of your post lab for this week. And I mean, So let me clarify. You're doing this in you can do this now even, right? In the remaining time that we have post lab. From what we would have expected to do today, but essentially do these critical thinking questions, submit them or send them to me via email over next week by Thursday 1 p.m. or Thursday noon. So send them to me. The critical thinking questions, your post and pre-lab, pretty much from week 4 to 5, with all of these questions and things that we've discussed. Consider them part of your post-lab. Um, Interpretations as well. These are things I've talked about. I also want to talk about transformation efficiency. This is important. Some of you have asked me about this. Charis actually asked me this, Charis, and let me just clarify this for everyone. So based on the number of transformants, it's shown this in the uh, in the manual, but essentially you're dividing that by pGLO's concentration, which in our case, pGLO, I don't want people getting confused, but write it down in your notebooks, 0.005 micrograms per microliter. So that's 0.005 micrograms per microliter. That's the concentration of pGLO. That's the concentration. And I'm going to try to answer your questions. So finish your question and send a post lab. All next Thursday. All of them can be. I was suggesting. Actually, let me answer questions at the end, Melina. Forgive me. I want to make sure I don't miss anything. And then remind myself if there's any outstanding items to discuss with you all. Okay, street plates, plaque assay, original titer, talked about that. Let's see, anything else? Do you see evidence? So, yeah, okay, I talked about this. I believe I talked about all of this. Let's see. What information does LB... Based on what you see here, essentially follow through with answering the questions that I've talked about. Beta-lactamase, you can look into the future. It's a bit separate, but essentially it's part of the future antibiotic susceptibility test assay that you'll be doing. So don't worry. I mean, like, do try to answer the question because you also have the manual to see with the details. But essentially, have a look. Understand the function and importance of the methods. This will help you uh, as well. Calculate transformation efficiency, counting the colonies divided by 0.005. And multiply that by the volume because technically, right there, it's per microliter, right? So nonetheless, before I move further, there are like questions as well to help prepare you, right? I know it's a lot of questions, but these are questions I want you to answer during reading week. And you could do this individually and submit them all to me next during reading week. Please do it by Thursday and let your peers know as well. Any of you that have contacts with other members that haven't attended today's practical or attended the seminar, please make sure that they also complete all of these questions, or at least the questions that I've talked about thus far. And then as groups, you can talk about like the reflective practice. Reason I say this is because these questions, as is shown here, they're going to help you individually prepare and focus on understanding the material before the lab exam. So think of this as your post-lab, before the lab exam, right? So these are things you need to individually understand and be able to answer. And so I want you to all submit them to me individually. But for the reflective practice, still in your lab notebooks and still through your submission, you can work as like talk through your groups. If you have their contacts, you can share them also at the end of today's practical with each other by PMing. But essentially, just feel free to answer these questions as groups if you're finding difficulty answering them. Some of them also connected things in the past. So think about how things reconnect, interconnect. and intersect with one another. So hopefully you'll be able to be well versed and prepared for not only the final, not only for your assignments, but also lab exam and for your future, careers and aspirations. سبحانك اللهم وبحمدك نشهد أن لا إله إلا أنت نستغفرك ونتوب إليك. So with that, I'm gonna answer outstanding questions now. Let's see. So Karis had a great question. For exercise 1, would you be writing the intro and conclusion paragraphs on the same semi-log paper we'll draw? So great question. Because give me one moment. I'm thinking. I want to be articulate in the words I say. They're separate actually. So reason is because exercise 5 for your post lab is going to be done and shown in reading week, whereas the deadline for like the generation time, your semi-log paper, the graphs and the results, all of that data is done separately. Yeah. Does that answer actually your question, Kiris? Essentially, intro, conclusions done in your manual, or sorry, in your lab notebook, just like as you would do regularly with your figures, your results from the lab itself. But then for the generation time assignment with the semi-log paper, that's actually going to be given separately at a separate time for pretty much the tasks that are mentioned here. I mentioned them as well earlier. Yeah, oh, sorry. To clarify for everyone, 4 curves, microplate, and sorry, yeah. My mind is thinking about a lot of questions actually. So finish the reflective question. Yeah, to answer your questions, yes, Melina, yeah, that's exactly what I wanted to answer, at least for your reflective questions. Pretty much slides. Okay, pretty much what I've talked about today, and I'll summarize it. Just you can record it and I can go back if needed. Generation time questions. Actually, before this, results. Your results, sadly, we don't have any to share today, but like pretty much you saw the ones I had from the Quarkus module, but from generation time, I want you guys to prepare yourselves, right? Preparing to include your pre-lab. So treat it as normal. Your post-lab material of the questions from your results from last week, right? All of that would have been done from last week, right? And the pre-lab material of generation time, etc. Now I'm just thinking in the flowcharts, but from okay, let me see. Apart from those things, um, okay, okay, I got it now. So reflective questions to answer the ones your first question about reflective questions. Pretty much everything from here on, slide 14 and downwards, all the way to 19, all of them you're submitting to me by Thursday next week by noon, inshallah. The difference for slide 19 is that one you could do, so all of them from 14 to 18, do them individually. Do your best to do them individually so that way you're prepped for your lab exam, and or at least you're able to sufficiently do your best to complete them in groups, in your groups. If you have contact with your groups now, like from those that are present here or outside, you can essentially ask them to you could work as peers to answer the questions shown here, the last 4 questions of slide 19. Does that answer your question, Melina, or am I missing anything? Maybe that answers it. Okay, perfect. Now, here's the other thing. Actually, just to conclude, generation time I talked about, pretty much preparing you guys with what you need to expect for 2 weeks' time from now. The sample results I talked about as well for conjugation, the plaque assay, preparing yourselves. If you want preparation, start preparing the formal lab report with your intro. And I don't know if you have methods. Um, I don't think there are methods. If there are methods, include them based on what you see from the PowerPoint, not PowerPoint, from on the module section. But essentially, prepare with your intro. Start at least, you can get ready the plaque assignment because there are things that I've talked about, right? Like being able to write down the PFUs, right? So hopefully you'll be able to at least, think about it. Conjugation, I talked about, there isn't a mini lab report unless mentioned on Quirkus. Otherwise, transformation efficiency, pretty much these things that we talked about, subhanAllah, all of these things are pretty much what I was just saying. Be able to do what you can from what we've talked about, from what I've discussed thus far. Streaking, yeah, okay. I think that should be it. Um, yeah, okay. I'm done. Any more questions?
Yes, I do have a question.
Yes, Karan.
Just to clarify, so you want all the questions from slides 14 to, well, the reflective questions on 19 to be answered on top of the questions from last week for the previous lab
So,
to be
great
submitted to you
question.
by Thursday.
Great question. Technically speaking, both of them could have actually been done for before today. Essentially, based on because these slides were already posted, right? And all the materials already available. I'm just like reiterating them for you. But technically, all of the material from last week would have already been done with the pre-lab, including the discussion and figures that you would be able to show me. So maybe to like clarify it for you guys, pretty much you want to include everything that I've talked about. Um, Sorry, something came my mind. Pre-lab, so post-lab of week 4, all of the figures, captions, remember they need to be detailed, right, from your figures. Um, I mean, pretty much you had the counting the CFUs from the spread plating, but essentially include that, include any other questions from the post lab, right, from the lab manual, the slides. That would have already been done for this week, but you're gonna show it to me next in your sorry, you would email your photos of your manual, sorry, of your notebook to me in on Thursday. So essentially you have you would show me your post lab, your pre-lab from this week, right? And then for your discussion points, pretty much include what you have here and anything else that I've provided. Do your best, the bare minimum, or actually, let's see. I see another question. Are we answering these questions in our notebook?
Uh,
Oh, that's a good question. You could, I would prefer notebook actually. Reason being is because um because I need to be able to I actually want you guys to be able to have them organized in your notebook so that way when you're preparing for your lab exam, you don't have to essentially go between multiple outlets to essentially prepare. So do them in your notebook. That's my recommendation. And pretty much there's just those 3 things that you need to do send to me your pre-lab for this week, post-lab from last week, results, figures, discussion, questions, and discussion points, slides, and PowerPoint or manual. So your pre-lab, post-lab, and then the questions for this week. And any
no,
other
sorry.
components that I've talked about thus far. By the way, next week I really hope to be able to send you guys the results for the microplate reader. So that way you can already be ahead for the generation time assignment. Because like myself, I want you guys to be ahead. I don't want you guys to feel like, oh, you're tense in a moment. I don't know what to do. And like I don't want to be penalized, right? I want you guys to be well ahead and ahead of the game, right? So that way you can one day maybe even have, 1 hour left of the practical, and you can literally go home at 11 a.m. or go leave the lab at 11. So, yeah. Garvin, you had another question.
I was about to ask for like questions that we need our results to answer. So I like for the streak plating, it asks, did you get isolated colonies? Is there any evidence of contamination? I can't really answer that right now.
Yeah,
So
that's okay. If there are components you can answer, do them sincerely, the ones that you can actually. But apart from the one based on what I've discussed today, right? Based on limitations that could exist. But apart from them, do all the other ones that you can. Like do everything that you can based on what you have available, based on what I've talked about, right? From my previous experience, based on the slides, based on the manual, based on the PowerPoint even from before, like the details that I've talked about. Hopefully, you should be able to complete them, even literally by this weekend. But I'm giving you guys until Thursday, noon, so don't worry.
okay.
Sorry, any other questions? Am I missing anything from any of you? Any other? Okay, let me just see if I have anything. Um, an alternative idea is if you want, you can also stay in, like, I could, like, if you want to actually spend this time answering questions, like right now, for the next hour, you could literally do so and literally save yourself the time of answering them next week. And then once you're done, you could just take the photos of them and then show them to me. Um, you could send them whenever you're done, essentially. I have another member who just joined. I think I know, but essentially, go over with. I talked about essentially three core things: the slides from this week. I talked about the results, sample ideas, results from last year. So you may already be familiar with those results, inshallah. And then the reflective practice and going through generation time, talking about formal lab report for the plaque assay. Um, this can also help you. By the way, these slides are very beneficial, or at least should help you not only answer through critical thinking questions. Sometimes you think I know there are a lot, but or they could be dense, but think about them critically, and they could really help you and prepare for the post lab, the lab exam, your FLR, and down the road, right? Even maybe your lecture material, right? So I'll just have a look if there's any ones that are concerning here. Um, based on lecture our slides here. So question one growth curve was constructed from data. This is so hopefully you guys should be able to see this based on actually lecture material. This is more easier to understand. Lecture question one. There is some relation to the lab, but most of that material that relates to it is actually in future weeks. So connect to the lab material. Anoxic is like limited or actually no oxygen available. So think about it like that. Some bacteria, are anaerobic, so you could also think of this. So maybe have an idea. So, okay, what is E. coli? Is it anaerobic? Is it aerobic? Is it potentially having both? No problem if you have to. No problem, Kiris. Have a great day. No problem. No problem. Generation time. I'm just gonna answer the other or go through others with you guys, providing some tips. GT for E. coli was determined. Okay. To be 30 minutes. Okay. Um, and that's actually something to think about. 20 minutes is the close condition. Think so, subhanAllah. Dr. Burns is giving you guys hints. No problem, Melina. Now I just want to let you guys know, think and answer these questions critically because these are hints for you guys to prepare you guys for the lab, and the formal lab report as well. Question three, let's see. Actually, before I go, before I stop, are there any final questions?
Final
Any?
question, sorry for me.
Yes,
I missed what you said about the microplate. Sorry, yeah, microplate? Microplate. For the microplate reader, does it have an built-in like incubator that's keeping the plate at like 37 degrees or
great question. So it actually does, but it has to preheat. So it does take a bit of time, about like within that 15-minute range to set up. So from Tuesday, like essentially it does, to answer your question. And which is nice because it's more automated, and so you don't have to worry about going in and out trying to take the spec readings from like outside of the water bath at 37 degrees, and then putting into spec 20 and redoing the process every 15 minutes. Believe it or not, this is what was actually done last year for the Spec 20 results. So you guys have it easier.
yeah.
Alhamdulillah.
Also, last follow-up before I go.
Yeah.
So for like how do I put it? Like for air absorption or air saturation for wells, what's a good like volume for like just having diffusion air saturation rather than needing a shaker as we do with the flask?
That's a good question. It depends. So depending on the flask, this is by the way, for those wondering, it's like a tangential, it's like a separate question. Because technically speaking, all of our flasks are supposed to be in under aeration. Even I believe the microplate readers should be. Aeration is like
Oh, wait, the
shaking.
plates are like under air shaking aeration too?
I believe so. I
Oh,
believe
okay.
so. So yeah, it's pretty interesting. But essentially, aeration is like shaking. They're like vibrating in and out. So I would just say keep that in mind, folks. Uh, and let me see. Okay, I think that's good. Okay. The other thing, yeah, take care, Sahar. Take care, Chana. Uh, um, you had another question. Oh, the volume. So different flasks have different like maximal volumes. There's optimal volumes that they would have. Let's say at 200, like a 500 milliliter beaker, for example, maybe at like 300 microliters. But itself, it's like the maximum would be was 500 milliliters, but it has like essentially that 500 milliliters label or increment is much lower to how big that beaker is. So for example, I have like a bottle here, and this is like I don't know if you can actually see it on Zoom, but like if I have
I
a
can see it.
bottle, you can see it?
Yeah.
Perfect. So
Well, you
like
moved it up. Yeah, there we go.
perfect. So like the idea is like there's like an edge where like it's like especially with regards to aeration because obviously it's shaking left and right, forward and back, and diagonally perhaps. The idea is that like there is like a volume where especially you want to reduce the risk of actually liquid seeping through any cultures as well. So essentially likely before the neck, at least for these trigonal beakers you generally speaking want to have the maximal volume is likely under this, likely even two-thirds. So likely if you see like my label right here on the screen, it'd be right
Mhm.
around two-thirds. Does that answer your question?
I think so. Actually, no, it's still is there
Okay.
like ever like a volume where it's like it's too much? Like I have a vat and it's like like waiting on air diffusion to like get this, properly aerated so my aerobic bacteria or whatever I need aerobically respirating is properly saturated. Is like there like a volume that I would be like, no, this is way too big. I need to do some like high pressurization thing or something to like get like proper air saturation.
That's a great question. So it does also like there are a few factors to take into account. One is indeed the pressure as well as the volume that you're working with, because of course, like now it's also like you understanding logistically why would you want to have essentially an entire flask, let's say, filled with bacterium to appropriately aerate the bacterium. Because the thing is, like technically speaking, if you have like a fixed volume, you could separate them into two flasks and essentially aerate them both accordingly, right? Which will actually increase the aeration efficiency. But Yeah, I would say like you don't want to overdo it essentially because if you have so much volume, you're increasing the pressure and that reduces the amount of aeration flow. That sorry, it essentially it wouldn't be the most optimal way in
All
simple terms.
right, thank you for your time. I gotta go.
No problem, Garvin. Take care. Any other questions, anyone? Even like marks wise, are there any
I have a
questions?
question.
Yes.
The log sheet, where can I get it?
Great question. So let's go on Quirkus. I don't know if Dr. Brun has posted it already. If she hasn't, then I will show you. Maybe I should send it to you guys. Once I get access to the microplate reading results, which just give me a bit of yeah. So it would be essentially like here under lab assignments. I don't know if you guys have this here because this is an older addition, but essentially it's like the figure that I had before. If you remember, I was like going from 10, 9, 8, 7, 6, 5, 4, 3, 2, and then goes just up to 1 because it essentially reconnects for longer graphs.
Thank you.
Um, yeah, I could also, let's see. I'm just imagining. I also don't want I don't wanna overdo it with I don't want Dr. Brent to be concerned, right? Because like she probably there is a pace that she wants to go forward with sending or like maybe like uploading the material. But let's see. Does that help answer your question? You can also print it online. There definitely should be like sample semi-log plate semi-log paper template. Even if we do it now, like
Thank you.
no problem. Yeah, semi-log paper online or PDF, both are printable, should help. No problem. Any questions? By the way, this is a
So,
separate point, but I just want to let you know some students have asked me, and I'm grateful, but like some people ask about like reference letters and stuff. So if you guys like are dedicated students, sincere students, authentic students that really want support and have shown commitment in the lab, just let me know at any time. More than happy to help. Sorry, yes, there was someone who spoke.
yeah, sorry, I was just going to confirm for the log paper. So, since we're not able to make it in today, all you need us to do, aside from like the post-lab questions and last week's stuff, we need to print the log paper and then just wait for the data to plot it, and then we'll bring it on week 6, right?
Yes. Give me one moment. Technically, what would you need a semi-log paper for now though? Because like the slide itself, I don't think you need like for the questions, is that what you're referring to? Or
No, just like separately, because you said that there's data we'll need to plot and submit.
yes. Yes. So once the results, once I get those results, like the microplate absorbance values, and confirm if needed. Or actually, once I get those absorbance values, I'll send them either on Quirkus or on the inbox, and with the semi-log paper for you guys to print out. So it will like I won't leave you stranded.
And will we need to submit that by email, or will that be an in-person thing?
That can be done in person. That you don't have. That's not part of today's because you actually need to have also the viable counts, which technically would have been done, but essentially you'd have to wait for those. Um, Essentially, you wouldn't have to do it now, or like you wouldn't. You don't have the results as of yet to complete them, but soon you would. So I would say wait until reading week, maybe around reading week. But I'll likely give updates, so don't worry too much about it. The main thing is pre-lab, post-lab discussion questions from here for
11
Thursday. Yeah.
post-lab. Okay,
Uh,
so don't worry about the log paper for now.
yes. But not for the. By the way, this is not for the lab report. Lab reports plaque assay. Sorry, I heard something report, so I hopefully I didn't hear you. You say something else?
Oh no, I meant report as in like reporting the graph paper too, not as in like the separate lab report.
Oh, yes, yes, that's correct. Yes.
Okay.
Any other
Also,
questions? Yeah.
sorry, and you said this last hour could be used to finish the questions now, so we don't have to do it later?
Yes, yes,
Okay, so this will still be open till 12?
yes.
Okay.
If you that's if you want to, because technically it's also on Quercus, so I don't mind if you want to like do it essentially independently and then send it to me around 12. That also works. I actually wanted to maybe save this more like for questions if people had any outstanding ones. Um, Give me just one moment, everyone. Okay, take care, Zara. Take care, Fatima. Take care, Funmi or Funmi. Uh, Wayakum, Hajra. Take care. Uh, Anyone else? Anyone? Or sorry, any other questions? Oh,
No, I
there's
think
one.
it's just me realistically.
Yeah.
If you want to save yourself time, I'm okay to just go and let the meeting end.
No, actually, if you have questions, I mean, you did come at the beginning as well. So I really appreciate it, Carlos. Thanks a lot.
Of
Or
course,
like
I appreciate you holding this. I'm just uh trying to take advantage of the time that I have.
that's so you know what? Let me see. There is something that came up a bit personally, so I may have to go, but I will at the meantime, if there's any outstanding, um, give me actually a few moments if that's okay. If you have questions, you can write them down. Actually, I don't mind to answer them if I have to go briefly
I
off.
can just summarize in like 2 minutes, and then if that's okay, unless you need to like run right now.
No, do you have the questions already?
It's just like it's a general thing, just so I can leave on like a good note. So I've already done
Okay.
the post lab for week 4, which is what you were asking for. I did the
Hmm.
pre-lab for ex sorry, week 5, which you also asked for. The last thing is just the post lab for week 5, which was slides 14 to 19.
Yes.
And that's it. There's no like discussion because we don't really have results to go off.
That's correct. I mean, if you want to discuss things that I've talked about, like about limitations for conjugation, that could work. Um, because, yeah, like I would say go with like the semi-observations that you saw from me, like going to different screens. And even if you want, like you could like I would say you could discuss things that you may expect or like outcomes that you would want to see essentially. And if they didn't appear, why is that the case essentially? But I would
More
say
like,
sorry.
sorry, more like predictions is what I'm hearing.
Um Quite, yeah, pretty much, yeah.
Okay, like an educated prediction. Okay.
Yes. But apart from that, like there are quite a lot of questions here, so I don't want you guys to get overwhelmed. I mean, if you want as well, like apart from this, I didn't make mention of it, but the if there are extra discussion questions in the notebook, the manual, then those could also be included.
Okay, sounds good.
With that, thanks a lot, Carlos. If there's any, I think I can end off here. Um, if you want though, yeah, I would say let me know if you have other questions at any time.
All right, sounds good.